Thank you for that introduction. So I realized this is the first one, so I'm completely open to experimenting with the tool, Zoom, and with different possible technical difficulties. So if during this talk anybody has trouble hearing me or um, any of the slides are not displayed correctly, please let me know, because I may not know. And so thank you, Tom, for inviting me. And really, this is a very unique experience for me. I'm very happy to see so many of you that I've known for so many years. And now that we're no longer separated by distance, as it were. In recent years, I've been really invested in non-invasive imaging of the brain, particularly looking at functional and structural connectivity in aging. And today I'll present some of our most recent work on the topic. Yeah, okay, so I guess I just got started. So resting state fMRI is commonly used to measure functional connectivity in the brain, particularly in the aging and patient populations. So just a brief overview of the concept. So we have this bold fMRI signal fluctuation that we then used to assess synchrony across the brain. So this was the first instance in which connectivity was measured just by looking at the correlation coefficient. As you can see, so on the top plot here, the correlation is high, is 0.84, and this is understood or interpreted as being a strong connection, whereas the bottom plot is the opposite. So using this very simple approach, we have been able to map a whole series of brain networks, notably the visual motor, as well as the attention and default mode networks. And all of these have been heavily used in the study of aging. So this figure I really love. I stole out of Tom's paper from 2013 Neuroimage. Really, a lot of my research really has been kind of surrounding this figure. So when we think about the origins of the resting state functional MRI signal, if we consider the outset, we are dealing with neuroactivity. It is some sort of buried in there. It goes through this hemodynamic modulation. And then we get sort of the hypothetically clean bold signal to which noise and artifacts are added. And then we have this resultant bold signal. So this should be very familiar to many of you. This is what I've been working on since the days of my PhD in some sense. But in terms of connectivity, we're looking at synchrony across two different regions. And if we look at a different scenario with a different vascular modulation, this is sort of modified from Tom's figure, then the time series for bold will be different. So then, although we have two identical neuroactivities that are 100% synchronized from the outset, when we get to the bold stage, they're no longer synchronized. So you can already get a sense of where the need is to assess the vascular influence on the bold signal, right? So in terms of aging, there has been quite a lot of research using functional connectivity measured using resting state fMRI. And by and large, the finding has been reduced functional connectivity, whether it's within the visual network or the motor network or the default mode. Actually, the visual network happens to be an exception, but the motor network and default modes and attention, those networks that I mentioned earlier, there have been observations of reduced functional connectivity. Interestingly, there is more of this recognition that some of them may be driven by vascular factors, including vascular reactivity. So this kind of preempts our investigation today, in which we look at what's happening to the vasculature in aging. So a lot of my work has been trying to assess the effect of vascular reactivity on resting state fMRI and the resultant functional connectivity measures. And we know also that vascular stiffening is something that is very common in aging, and that can very likely lead to reduced vascular reactivity as we measure it. So if we look more closely at what's happening in vascular stiffening, or commonly known as arterial stiffening, we can see there are two major causes. As individuals age, there's a decay in the elastin, and there is an increase in the concentration of collagen. So these two factors, which pertain to the vascular wall, are recognized as causing vascular stiffening. And risk factors, let's face it, these are all very common in aging as well: hypertension, diabetes, metabolic syndrome, obesity, clinical obesity, and as well as hypercholesterolemia. These all contribute to the likelihood of vascular stiffening. So this, when we look at the generalizability of this relationship, arterial stiffening can be a cause and an effect to atherosclerosis, and vascular aging is sort of a more general term that applies to this and other types of vascular changes in aging. But arterial stiffening can actually precipitate the accumulation of plaques, which actually lead to atherosclerosis, and it's characterizing that condition. So with this in mind, we think that although we study largely healthy control older adults, a lot of them do have these conditions. I think the majority of them do have some sort of hypertension at the very least. So what then can we conclude by looking at sort of common age-related vascular conditions versus function and cognition as measured using fMRI? So this is a paper from a while ago, 2004, looking at the association between the beta stiffness index and working memory. So you can see this sort of is kind of in agreement with what we would expect. Working memory intensity as measured using fMRI goes down as the stiffness goes up. So in the animal model, we try to tease apart the neural and the vascular aspects of it, because even though we're measuring working memory, it could be that the vascular changes led to the neuronal changes and so on. But if we are looking at the animal model, there has been this paper from Frederic Lesage's lab in Montreal, where they looked at inducing vascular calcification, so artificially introducing vascular stiffness, and then looking at functional connectivity. And they do see in all the networks they looked at, functional connectivity went down due to the calcification. So there's not necessarily any neural change. This is all vascular, right? So could it be that the same is happening with our study of aging using resting state fMRI? So this is a recent paper that we came out with addressing this question. So our hypotheses are that first, vascular stiffness does have a significant impact on functional connectivity measures in the aging human brain. And secondly, this effect may contribute to the observed age effects. So this was led by my student Ahmed Hussein. So we measured vascular stiffness using pulse wave velocity. So this is sort of a little animation demonstrating what it is. This is basically the velocity of arterial pulse wave traveling between 2 points, in our case, the heart and the brain. And so you see as the pulse advances towards the brain, a part of it propagates to the brain and another part reflects. But as the vascular becomes stiffer, the velocity of that propagation will increase. And so so the vascular is less able to absorb sort of the shock, as it were, from the cardiac pulse wave. And so this is what happens in aging. So by that, I mean, if we have a higher pulse velocity, that is indicative of higher stiffness. So this has been used for many years. And there are two main ways in which pulse wave velocity is measured. One is carotid femoral, and another one is aortic. So in the case of aortic, which is the one we used, we measure the pulse wave at 3 different points along the aortic candy cane. So as you can see along here. So this basically gives us these multiple time series, as you can see here, and we just have to measure the time of arrival of these 2 different boluses, and we know the distance between them, so there we can calculate the pulse wave. This was done using cardiac phase contrast imaging. So in healthy controls, we see the pattern on the left. This is sort of in contrast to the pulse wave pattern for those with arterial stiffness. You can see this increased velocity and increased pressure. So this is something that happens with aging, but it also varies with height and heart rate variability. So there is that caveat that those who are taller tend to have higher pulse wave, but at the same time, our driving variable in this case is age. So our question is, can this effect, which is largely considered systemic, affect the resting state fMRI measurements? So our data was obtained from the Toronto transgenerational brain and body study. So it was data from a 1.5 Tesla scanner involving 59 healthy adults, evenly divided into young and old. Actually, these are sort of bigenerational cohorts. So the young are actually the offsprings of the older. So this is sort of how this study was designed. We saw the opportunity to study the effect of blood pressure and vascular stiffness. So pulse wave velocity was measured using ECG-gated phase contrast MRI along the aorta, and functional connectivity as well as cerebral blood flow were both measured using pseudo-continuous ASL. And in terms of aortic pulse wave velocity, it was a standard toolbox called segment. Functional connectivity, this is sort of worth a little bit of mention. We did not look at network-specific connectivity. Instead, we looked at network or seed independent connectivity. This is a metric called the intrinsic connectivity contrast. So what it is basically a weighted average of the correlation coefficient from a given voxel to every other voxel in the brain. So then we will repeat that for every single voxel. And in the end, we have this sort of intrinsic connectivity contrast map. And finally, we also looked at the amplitude of resting state fMRI signals. We call that RSFA. Okay, so to now dive into the results. So first we looked at the effect of pulse wave velocity versus the RSFA, so the signal amplitude. We see that the blue represents a negative association. So increasing stiffness is linked to decreasing pulse, both signal amplitude. So this is actually expected based on what we know about stiffness reducing vascular reactivity and vascular reactivity driving the RSFA. So this is to be expected. But look at where the blue regions are. A lot of the regions, for example, here the precuneus / posterior cingulate, and here the superior parietal gyrus, they are part of the default mode network. And the default mode network has been heavily studied in aging. So how does this translate into functional connectivity measurements? So again, these regions here, associations between arterial stiffening and functional connectivity. And again, you see that there's a negative association. So stiffer vessels are associated with lower functional connectivity. Here, we're not considering age at all. We're simply considering arterial stiffness. And so you can see again here, the precuneus / posterior cingulate lights up again. This is also an area that was influenced in terms of the both signal amplitude. If we look at how these two overlap, so the two maps that I showed previously, so the green is the overlap. I mentioned this area, so you can see this is an area that overlaps between these two types of associations, meaning that this green area demonstrates lower both signal amplitude with higher vascular stiffness, as well as lower both functional connectivity with higher arterial stiffness, but this doesn't generalize across the entire brain. You do see a lot of blue here that has no overlap with the red. So there is a lot of uniqueness to each type of association as well. So our next question is, how does this resemble the effect of age? So are these the same as the effect of age? So we went on to look at that in our 59 subjects. Increasing age is linked to decreasing signal amplitude. So the RSFA blue, I do get the sense, even if the figures are not side by side, but the patterns are different between the effect of age and the effect of arterial stiffness. But they are both in the same direction. So now we will try to overlap those two as well. So the green represents regions in which the arterial stiffness effects on the bolt signal overlap with the age effects on the bolt signal. So again, so this midline here, which is the hub region, the posterior cingulate, it shows up again as being both affected by age and arterial stiffness. But there is also a lot of blue here, which is only the arterial stiffness effect, which doesn't show any effect of age. So there's some uniqueness to the effect of arterial stiffness. It does not explain all aging effects, but it does explain aging effects, at least as it relates to the default mode network here. So next question, we wondered if vascular stiffening was not a large vessel phenomenon, because we are measuring it from the aorta. We're not necessarily measuring it from the microvasculature. You might ask, so because the buffering mechanisms, as the wave enters the brain, it should be dampened to the degree that it doesn't cause any vascular damage. It doesn't propagate that far, but that's not the case based on what I've seen in the literature. It's definitely a case to be made for the microvasculature to be the most vulnerable to undampened pulse waves coming from the heart. So we have our macrovascular stiffness measures, and we try to look at the dependence of our previously observed bold signal effects on the size of the vessels. So to do this, we got this the large vessel atlas. So the red here, you can see are sort of voxels demonstrating high probability of finding a large vessel across multiple individuals. So in this case, 69 individuals. So you can see here, this is the circle of Willis, and up here, this is the sagittal sinus. So these are all well-known large vessels, whereas the smaller vessels, which you don't see, they've been thresholded, so they're not shown because there's more variability across different individuals in those cases. So we're using this as an indication that, okay, high probability of observing it in the same location means that it is a larger vessel. So if we were to plot the probability of large vessels against the strength of the association between the bolt signal amplitude and arterial stiffness, so we see a negative relationship. Let me just pause a little bit to kind of explain this, because it may not be apparent, but on the y-axis, so the higher the value, the stronger the association between the bolt signal fluctuation and arterial stiffness. But if you look at how it's progressing, so the regions with the largest vessels tend to have not to have the strongest association between bolt and vascular stiffness. So by elimination, we concluded that, okay, so the strongest associations between these two things are actually in the smaller vessels. So that's sort of how we reached at that conclusion. But this requires also further investigation. So we have no reason to believe this is a large vessel phenomenon. And one more question, dodging, is because we are kind of paranoid about what we might be confounding. And we are here looking at the effects of tissue loss. So because this is an aging population, we expect there to be cortical thinning, which is sort of on the left. So cortical thickness versus pulse wave velocity. So the blue regions represent regions of thinning with higher stiffness. And as it happens, if we control for this, we do not see a change in sort of the kind of resting state fMRI versus arterial stiffness relationship. And when I look closely, I noticed that if you look at these two maps, they really don't overlap a whole lot. So it might be a reason why there's no effect of controlling for cortical thickness. Okay, so to kind of wrap up that portion, we found in this study that pulse wave velocity, which reflects arterial stiffness, also reflects arterial pressure and sympathetic vascular tone. So higher pulse wave velocity means higher stiffness, and it has been reported to account for up to 10% of the actual signal, but in our hands, it does have a strong and widespread effect on resting state both fMRI, whether it be the signal amplitude or functional connectivity. And our findings are actually consistent with the earlier animal study that I showed with the sort of calcification model. Okay, now a little bit of change of theme into structural connectivity. Is there any question related to that first part? Okay, if not, then I can continue. So structural connectivity is the other side of the coin. We have been looking at this in terms of how DTI is tremendously used in the study of aging. And most of the data that's out there is based on single shell DTI. So by that, I mean there's one single non-zero B value. And we use it to measure fiber integrity through mainly mean diffusivity and fractional anisotropy, MD and FA. And in aging, the primary observation is that FA goes down. So this has been interpreted as structural degeneration. And paradoxically, there has also been observations of increased FA with age, and this does not compute if we consider our traditional DTI model. So this is what motivated this other project here. So if we look at diffusion tensor imaging, so the single tensor model, we have a single diffusion tensor we're fitting here through which we derive the mean diffusivity and fractional anisotropy, right? So we can have a isotropic tensor in which the water is equally likely to move in all directions, or anisotropic tensor, which is more representative of the white matter structure. Now, in terms of measuring sort of structural connectivity, we usually fall back on FA. Sort of that's sort of the bread and butter of a lot of tractography algorithms. But we are affected by the ever-changing tissue topography in aging. So there's partial volume contamination as well. So some models actually do account for this. So when you're not assuming each voxel contains a single fiber or single tensor, you can incorporate an additional tensor, or you can have this ball and stick model in which you have an isotropic diffusion compartment and you have a directional compartment. So the stick here represents the cellular compartment, and the isotropic one can be sort of interpreted as the extracellular fluid. So at the same time, white matter also atrophies. So we do see whole brain white matter volume going down as age goes up. And this in turn is reflected in the increased CSF fraction, so cerebrospinal fluid fraction. And at the cellular level, so if you don't consider the CSF, if you don't consider the atrophy, if we look at each individual fiber, we do see that there's the increase in extracellular space surrounding the fibers. If you take these two voxels as representing the same area, but in the older version, there is more extracellular space and the axons do get bigger. So overall, there's a significant change in the actual microstructure, but we may or may not be able to see that using single shell DTI. So regarding this increased extracellular space that is not atrophy, that's simply spacing around individual fibers, we expect to see increased mean diffusivity because the myelin sheaths are expanding, there's more space in between them, and we do expect to see decreased fractional anisotropy, as I mentioned before. Right, so this is a schematic of a healthy, younger white matter voxel on the left, and as we age, we do see this increased gap in between fibers. That's what we expect to see based on animal studies. So a bit about the technique that we propose to use to separate out the cellular and the extracellular environments. This is a technique proposed by Alfred Pasternak in 2009. It's called free water imaging. So the draw of this technique is that it does not require multi-shell DTI data. Supposedly we can use regularization so that we can estimate two parameters from a single measurement based on the regularization. So here we have the diffusion tensor. This is 1 minus f representing the actual cellular compartment and the extracellular compartment, which is free water, f. So these are the two things we're interested in. So this was, the work I'm going to present today is based on a recent paper by my student, Ph.D. student Jordan, and he's looking at the effect of separating the intra and extracellular compartments. And this was really also a motivation factor, this paper by Miller and colleagues in Nature 2016, using a sample of 5,000, looking at the effect of normal aging. And they noticed this increase in FA with age, this red here. So this was counterintuitive at the time. We thought that we are only going to see FA going down with advancing age. But if we look at where these red spots are, they're really at a fiber crossing between the internal capsule and the cortical spinal tract. So cortical spinal going this way. So what could be happening is that in aging, there might be a selective degeneration of one of the fiber tracts. So not both of them are degenerating at the same time, maybe just one. So this is something that could lead to an increased observed FA. But they did have 5,000 subjects, and we noticed that other studies of the many DTI-based aging studies, none of the other ones reported any increased FA that we could find. So we tried to see if we can see the same thing with a much smaller data set. This is a data set from the MGH, 212 subjects. So with conventional DTI metrics here, including axial diffusivity, radial diffusivity, mean diffusivity, and fractional anisotropy. So as we expected, we do not see any increase in FA anywhere. But we do see this increase in diffusivity. This is all according to, in accordance with the literature. But are we measuring authentic fiber microstructure? By authentic, well, it's quote unquote. I use it to represent the sense that we want to measure cellular microstructure and not a mixture of cellular and extracellular, as I mentioned before. So young fiber tends to be tightly packed together, whereas older fiber tends to have more space between them. And we have these different compartments. We have the hindered compartment, which we categorize as cellular water, and we have this blue, which is free water. So free water, the definition is really, it's not a biological definition as it is the measurement definition. So it sort of gives us a sense of how much extracellular space there is. And we get a sense that increasing extracellular water might be the one that's driving the age effects on conventional DTI metrics, not so much the cellular ones. So we want to be able to separate the two. So I mentioned before the partial voluming with CSF. So there has been a lot of work looking into this, and I cite a few papers here, but I want to distinguish between that and the extracellular space. So you can see here is a schematic of crossing fiber structure, so hypothetical crossing fiber. The primary fiber here is intact, and you see the tight packing. And the secondary fibers, you have two possible scenarios. One is a scenario in which there is no apparent white matter volume reduction, but there is an increased interaxonal space. So this is increased extracellular space that fills with water. Okay, so this is part of demyelination because then the fibers shrink. or the spacing in between the myelin sheaths, they increase. Whereas once there is a time point sometime in the future down the road where the fluid can actually come out, the interstitial fluid can exit from that interaxonal space into the CSF, then you would see a white matter shrinking happening, but not before that, right? So I want to distinguish between extracellular space, in which you are only looking at the water that's not in the myelin, that's not in the axons, versus cellular space, in which we're only looking at water that's within the axons and myelin. So we expect extracellular water to increase with age, and cellular water to also, cellular diffusivity to have two different directions. So along the direction of the axons, because of age-associated debris accumulation, we may see reduced axial diffusivity, but along the direction of the myelin sheaths, radially penetrating the myelin sheaths, we do see, because the packing of the myelin is looser, we do expect to see increased radial diffusivity. So the two combined gives us sort of this increased overall mean diffusivity, but really when we're looking at conventional DTI data, we're looking at a mixture of these two spaces. So I just want to make that very, very clear at this point. So how do we propose to separate the two spaces? I mentioned free water imaging before. So these are some of the papers using this technique. It's becoming increasingly adopted. And we're proposing to separate out the cellular and the extracellular compartments. And the questions we want to answer are, one, if we account for extracellular contamination, can we uncover cellular FA increase in aging in a smaller cohort than 5,000 subjects? And two, what does FA increase in aging really mean? Do we really expect the tissue to get better and more healthy with older populations? So as I mentioned before, 212 healthy subjects, and they're ranged 39 to 92 in age. This is from NGH Siemens Trio 3 Tesla scanner, 60 directions with a single B-value of 700. And we also acquired anatomical for reference. And our methods are basically, we use conventional DTI, so through FSL, and we also use free water imaging modeling with the extracellular and the cellular compartments separated. And so these are the symbols representing the corresponding DTI parameters for the cellular compartments. So I will use these from this point forward. So F is the extracellular free water compartment, and then anything with a postscript T means it's the cellular compartment. To summarize on the whole brain scale, so white matter volume does go down with increasing age, and the mean diffusivity in the white matter skeleton also goes up in increasing age. These are not surprising, but we did see that free water, as we measured using free water imaging, also went up with age, and it was actually associated with the highest R-square value of the three. Now, in a more spatially defined manner, these are the regions that are most affected in terms of free water per year. So yellow is higher than red, and by and large, the entire white matter skeleton experiences extracellular free water increase with age. If we now look at mean diffusivity, it also goes up with age, but if we separate out the cellular mean diffusivity, then virtually all the effects are gone. So this indicates to us that the mean diffusivity effects that people usually report are not so much associated with the cellular space diffusivity, as I mentioned before, but it's more an extracellular phenomenon. So spacing between the fibers getting bigger. Whereas FA, do we see an increase? Do we see that? That's what we were hunting for. And we do. Now, when we separate out the cellular and extracellular compartments, so for the cellular compartment, we do see in the same region that the other Nature study reported in before, this crossing between the internal capsule and the cerebrospinal tract, we do see that increase in FA here as well, but we were not able to see it before. So just to reiterate what we think is happening, there's only one most likely explanation is that there's selective degeneration of crossing fibers. So on the right side, you can conceivably entertain the notion that the FA there will be higher than the FA here, because in any given box, so these two populations are mixed together. So just to confirm that fact, we also ran bedpost X. So this is sort of FSL's tractography tool. So we are able now to look at the primary tract population versus the secondary tract. So you can see that there's no change in the primary tract in this little rectangle here, where there's a decrease in the secondary tract. So this sort of corroborates what we were thinking is happening. And finally, we wanted to see if this accumulation in the fiber debris is causing a decrease in axial diffusivity across the brain. And we are seeing that the blue represents that phenomenon. But we do see in the same fiber crossing region where FA was going up, axial diffusivity is also going up. And the contrary is happening with radial diffusivity, where we see increased diffusivity with aging everywhere else in the brain, except for in this region where we see FA increase. And then by the same token, actually, the radial diffusivity is going down. Really depends on which we're defining as being radial. So in this case, because the sort of partial volume effect between the two different fiber tracts is going down with age, this can lead to an apparent reduction in radial diffusivity of the primary tract. Here. So to wrap that part up, so with the free water corrected diffusion tensors, we see a very different picture of microstructural connectivity. Most of the age-associated mean diffusivity reductions are gone, or increases are gone. And we do see in some select regions increased fractional anisotropy, which had up to now been counterintuitive. So we can justify that as being due to the crossing fibers. And this is something that when we study aging or disease, we have to pay more attention to. And axial diffusivity decreases with age, but not in the crossing fiber regions. We expect in crossing fiber regions it's the other way around. So a lot of things are kind of turned on their heads when we have crossing fibers. This is something that we should all remember. Of course, if microstructural atrophy is expected, it is always important to evaluate the contribution of extracellular versus cellular water. So in DTI, this is, I think, always important. And if you've already acquired data, you can apply the single shell free water model to it, as we have. But if you have not yet, I think it would be more comforting if you were to acquire two shells. That way you don't have this ill-posed problem to solve, and you have two measures, two unknowns, and definitely model the tensor as sort of a tissue tensor and an extracellular water compartment. So take-home messages, basically everything might be wrong. Functional MRI-based connectivity are largely dependent on the vascular biases. So today we talk about vascular stiffness as one of them. And DTI-based structure connectivity also rely on the assumption of a single tensor. So the mixture of the cellular and the extracellular compartments is shifting with age, and that is something that's typically not taken into consideration, so it should be. So we're at the end of my talk. I just wanted to thank my funding sources and my lovely lab. And we actually do have postdoc positions available if anyone wants to live in Canada. And thank you so much for your time. I hope I didn't cut out again.
No, that was great. Thank you very much, Jean. That was excellent talk. So I think we're open for questions now. So we can do it in two ways. One is if you'd like to send a message to the chat, so you want to ask a question, but we're probably also a small enough group that if you just want to unmute yourself and chime in, that's probably going to be okay as well. So why don't I start off with one question? So Jean, that's very interesting in terms of the white matter and sort of selective degeneration of one direction versus another. Do you have some hypotheses as to why one direction would be more susceptible to degeneration versus the other direction?
Um, I do have one. So this is quite common in the study of aging. We say the last in, first out. So this has been the case with cortical thinning. So we do see this happening, having sort of an anterior posterior gradient, where with the diffusion maps that we got, that gradient was not as apparent, but it is somewhat there. So a case can be made that if the cortical spinal tract was one of the first to be in, the brain has more vested interest in it to keep it healthy, whereas the later tracks, which are associated with higher cognition, in the sort of evolution of things, they might be considered more dispensable. So that's certainly what we have been seeing with regards to this particular crossing.
Right, that's very interesting.
Does the data suggest that it is the cortical spinal that's maintaining the anisotropy, and it's the whatever's crossing that's not?
It does. That's the other thing. So it does come together. The sense that the cortical spinal tract is staying intact. I did not show the sort of the properly oriented figure here, but we do see that.
Another question is when you separate diffusion into an anisotropic component and a water component. So my understanding is that the numbers as far as how far water molecules actually move in the time frame of a DTI experiment, the water between the fibers, I mean, it should be bumping into the walls of the neighboring fibers quite a lot, right? In the 20 to 50 or 100 milliseconds. I think mean motion of water
Right.
molecules is like 10 microns or something like that, right? Which is still much larger than the spacing between those fibers.
Yes, definitely. So that's been the thought, actually, for in the DTI community for some time, that there's not enough space in deep white matter for there to be free water. So in the free water imaging model, we set a threshold for mean diffusivity. We set it to 3 micrometers squared per second. And it is actually at that threshold only that we call something free water. So anything below that doesn't qualify. Of course, there is the chance of artifacts being introduced in here. So we do have that same chance of running into noise-related biases everywhere else in the brain. So the absolute value of the free water fraction is probably hard to interpret, but it's mostly the age trends, sort of the changes with age that we're observing. Yeah, so we were surprised to find so much effect from the extracellular water in aging, but it is there in deep white matter. And I will also say you're absolutely right that within the typical diffusion acquisition parameter space, we don't expect the water to move very far. So this is something that depends on the acquisition protocol. So the mixing time, echo time. So these definitely would change the apparent F value, so the free water value. This is not something that stays constant. So we are actually trying at this moment to cross-validate the single shell DTI free water measurements with 2 shell measurements. So that way we have a better handle on whether these values are pure imagination introduced by the regularization method. Because with 2 shells, you technically don't need regularization. You're just fitting 2 unknowns to 2 measurements. So that work is ongoing, and I'll be happy to report back, because this is something that people have this skepticism, and we certainly had at the very beginning. I mean, I'm not done with being skeptical, of course, this is why we're working on it, but it does give us some interesting findings that we can kind of base on animal histological research. So there's something interesting there, but we should always keep an eye open for possible confounds.
Great. So I had a question in terms of when you're presenting the associations or I think slide 17, like with the resting state amplitude, it seemed like there was quite a bit of left-right asymmetry um, in this, especially,
Yeah,
maybe the more superior slices. Do you have any sort of thoughts on what might be causing that?
that's a great catch. Yeah, we spent a lot of time trying to figure this out actually. And I don't know that we actually did eventually, but we did explain it in the paper. But because there is this asymmetry in terms of cortical thickness, that's a naturally existing asymmetry, just like some are right-handed and left-handed. So there is this cortical thickness asymmetry, but then when we try to control for cortical thickness, we didn't see an effect. So yeah, so I'm not sure. This might just be in this population, but we also looked at the data points, and there were not any obvious outliers. So that's something that we're trying to explain still. We want to do this in a bigger data set. This is only 59 subjects.
Thank you. So feel free to chime in if you have any questions. So another question I have is sort of you've pointed out some of these issues with um, vascular effects affecting our resting state fMRI measurements. But most studies don't sort of do any measurement of pulse wave velocity or any other vascular measures. So what would you suggest for the field? I mean, we're gathering all this data, and what do we do with it if we don't know what these vascular compounds are?
Um, yeah, that's a great question. And this is the point of doing all this, but that's a question that sometimes keeps me up at night too, because there are all these data sets acquired, and should we just give up using resting state fMRI? Um, because one of the outstanding questions now is, are the vascular factors tied in with the neural factors? In the animal model, we can separate the two, but we can't do it in the humans. So that's something that requires some careful thought. I was hoping that I could get some of your feedback on that, because so far the best I can do is just to say, for example, if we're trying to assess functional network integrity in aging using resting state fMRI, I would also try to throw in maybe cognitive measures and neuropsychological batteries to try to justify in more form than one that these measures are reflective of brain function. Another way is if we were to use the actual resting state signal fluctuation as the surrogate, but there's going to be a lot of controversy over that because the signal amplitude is not purely neural or vascular, and it's not even purely a useful signal. It can have lots of artifacts in it. So unless you have a very large sample, in which case you might be able to get away with all the confounds. Otherwise, it would be a hard sell, in my opinion, to use that as a vascular scale factor. Although it has been done before, but it's not easy to justify fully amongst those that do research in this area. Yeah.
think
So
that
this is a
so
tough
you think
question.
if you looked at like the fluctuations in larger arteries or something or larger veins, here you did uh, you had arterial spin labeling. So there are some measurements. I guess the Cardiff group has sort of argued that you can look at some of the you know um, fluctuations due to heart activity in the smaller vessels to look at compliance. Do you have a sense of like if you were able to add one scan to every resting state fMRI protocol, do you have a sense of what would be the most helpful to add to sort of get the information that could help sort of us interpret what is vascular versus what is neuronal?
Um, so right, so if I were able to add a scan, so instead of using existing data, if we're going to just try to be optimal with new acquisitions, um, so our approach currently has been to acquire resting state and tidal CO2 so that we could also quantify vascular reactivity.
Mm.
But that's just, I mean, based on a given method, but that does not clear up the question of whether that vascular reactivity, if you regress it out, you throw out also useful information. That does not help with that particular question. So I think, yeah, so going forward, probably some kind of, not pulsatility measure, but vascular reactivity measure that's independent of the bulk signal, probably. Yeah, so that's something I would definitely consider, because based on a previous study that we did, looking at the influence of blood flow, vascular reactivity, and blood oxygen, their influences on resting state fMRI, vascular reactivity was the biggest one. So we need a true measure of vascular reactivity, but once we have it, then the question is, what do we do with that signal? Because a linear model in which we regress it out is probably not the right way to go. So there's always going to be that question. Aside from the scanning, there's that question of the analysis.
Mm-hmm. Yeah, thank you.